Okay.
Oh, all right, kids, it's time. Welcome to the first Bio 285 review session. I'm John.
And I'm Jen.
Yes, we have the same last name. No, we are not related. Important to get that out of the way. And so, yeah, let's get started.
Okay. Okay, so this is the structure of an amino acid. Here we have, can I move? Can I get over here? Okay, thanks. Okay, so we have an amino group. It's the nitrogen with 2 hydrogens, and you have that in every single amino acid, all 20 of them. And you also have a carboxyl group on the right side, depending on how you're looking at it. It's the carbon with 2 oxygens to the right of the central carbon. You also have a variable R group in every amino acid. They're all different, so the R groups are going to change for every single amino acid. This R group is going to characterize your amino acid. So its chemistry is going to be different, its size is going to be different, and it's going to affect the overall structure and function of the protein. And should I talk about backbone here or no? No. Okay.
All right. So formation of peptide bond, very important thing we talked about in class. So this is a dehydration reaction. And so if you look at the square / box in the middle right there, the highlighted atoms of 1 oxygen, 2 hydrogens are the ones that get taken out of the 2 amino acids and combined together into the peptide bond right there. We did talk about how the peptide bond is very specific. It is shortened in length because of resonance, and it has basic double bond structure. It is a stronger bond, and then that is very important for you guys to know, because that is why covalent bonds are the strongest types of bonds that we talk about in this class. Disregard inorganic chemistry right here. Like, we are not talking about that type of ionic bonds. As far as Bio 285 is concerned, covalent bonds are the strongest. And so opposite to the formation of the peptide bond, the breakage of a peptide bond is a hydrolysis reaction, which means you need a water molecule in order for that to happen. And so it is a very time-consuming process. And then there's also a lot of energy release when that does happen. All right.
Okay, polypeptide anatomy. Polypeptides are just composed of a bunch of amino acids that have peptide bonds. So you're always going to start on your N terminus, and you're going to label from then all the way to the C terminus. You're going to start with 1, 2, 3, 4. Numbering amino acids always starts at the N terminus. Your R groups are always arranged by chemistry and size. You don't want big, bulky R groups next to each other and interfering with each other. They're just arranged in a certain way based on their chemistry and their size. And like John talked about, peptide bonds have resonance, the double bond characteristics because of this carbonyl group. So when you carboxyl group, there's resonance between this double bond and then this bond with the nitrogen and the peptide bond. And yeah, that's just a polypeptide. It's just a bunch of amino acids linked together by peptide bonds. Anything to add?
All right, primary structure, the most basic structure of proteins, as far as we're concerned. And so it is just the sequence of normal amino acids in a protein. And so peptide bonds are what link amino acids together. And so your average protein is probably going to be ranging from 100 amino acids to 1,000 plus amino acids. So when Laura talks about like an amino acid that's on number 25, 30, that is very much N terminal, because that is the early side of the protein as far as synthesis goes. Again, peptide bonds are strong, and the lots of energy to break requirement is very important in order to hold protein structure together. Obviously, you want a functional protein. Each protein has its own specific function, and its structure is highly important in the maintenance of said function. And so covalent bonds being formed, forming the peptide bond, is what holds this potential protein in order to do its probable function.
Oh, sorry.
Oh, the slides will also be
Yeah,
uploaded
We'll send
and
them out.
the recording will be on YouTube later.
Okay, so secondary structure, you should always think of hydrogen bonds. This is your backbone atoms interacting with each other to hold the protein. There are no R group interactions when you think of secondary structure. These hydrogen bonds are going to happen between atoms who have high difference in electronegativity. So think of like the difference in electronegativity between an oxygen and a hydrogen is very large. So they're going to want to bond about, like in an alpha helix, they're going to want to bond about 4 or 5 amino acids away to give that helix structure. In an alpha helix, your R groups are going to be arranged on the outside. They're going to want to interact with the environment. They're going to want to make bonds. R groups are going to want to bond with different R groups with different elements in the environment that the protein is in. And then in a beta pleated sheet, the R groups are going to be interspersed. So they're going to be alternated. One's going to be up, one's going to be down, the other one will be up, and so on. And yeah, another like name for hydrogen bonds. The reason why hydrogen bonds happen are these dipoles that happen. So partial charges on certain atoms, partial positives and partial negatives will bond with each other to form hydrogen bonds. And that's all you should think about with secondary structure. No R group interactions.
I wanted to just emphasize something. Oh, good. So when you think about electronegativity, I mean, most of you guys have taken intro level chem, and I'm assuming a lot of you are in orgo. You don't want to think about electronegativity as just a number that atoms carry, and whatever. There's just electronegativity, and you think about like fluorine, and fluorine is like crazy. Just think about how attracted the electrons really are to these molecules. When a atom has higher electronegativity, all that really means is that the electrons are going to tend to hang out around them more. And so if you think about like a water molecule, like if you have seen one of those like fancy little like 3-dimensional like water molecules where the oxygen is red and there's 2 white hydrogens, all that basically means is that the hydrogens won't really have as much electrons hanging around them as the oxygen molecule will. Or not the oxygen molecule, the oxygen atom. And all it's saying is that the electrons are going to be more available to the higher electronegativity atom. That's basically all you need to know. It's not really like think of a number. I mean, I wouldn't necessarily think about it as a number because like a number doesn't really tell you anything. Like if Laura really wants you to like think about it in like a visual way, and if you think about like how like a heat map, like the red is like hotter and then blue is like colder. If you think about like red being higher electronegativity and like blue being lower, then like an atom that has higher electronegativity will be more red than the atom that doesn't have such high electronegativity.
Good.
All right.
Okay.
Tertiary structure. Okay, so this is where it gets interesting. So this is overall a 3D shape or the perfectly folded protein structure that we're looking for. And so it's just a polypeptide being folded in a way such that it can perform its function. Laura is a very she's a very strong believer in teaching you guys that protein structure is completely tied to its function. The second a protein loses its shape, it will not be able to perform its function. Even if it's just a slight bend, it will not be able to perform its function to the level it's supposed to. And so if you take nothing away from this entire presentation, I just want you to remember that protein structure leads to function. That is the number one thing for this slideshow. And so one very important thing that I want to talk about, tertiary structure involves R group interactions. Most of them are going to be non-covalent. And so you're talking about different polarity groups interacting, or you're talking about non-polar groups having, I guess hydrophobic effect is what happens when a protein folds in such a way that the non-polar amino acids go to the inside, so they don't get exposed to water. And so what we will also see is that when that happens, there's this thing called van der Waals interactions, which is just basically non-polar groups interacting with each other in a way kind of similar to other non-covalent interactions, but they're just non-polar. The only covalent interaction happening between our groups is a disulfide bridge. This is very important to write down. I'd like highly recommend writing it down. A disulfide bridge is when you get two cysteines and they bind together in a covalent way that leads to protein structure. So that is very important. It is the only covalent R group interaction, and it is formed to hold protein structure together. If you talk to Rosella about creatures that live in extreme temperatures, she will give you a lecture on how that is very common and present in their protein structures because they are able to hold structure for like longer in stable like pH levels and temperatures that other proteins will not be able to be stable in. But that's just if you're curious about it. In the meantime, just think about it as the sole covalent interaction between our groups.
And so you said that was said what was one said something earlier about the
Oh,
outside of the
protein structure leads to function. Yeah, that well, that disulfide bridges are the only covalent interactions between the two R groups. Yes.
So like with, like you're saying, like this, disulfide groups are really important for like keeping the protein like strong together.
Mm-hmm.
There was like a question on the pump set about like three cysteines. So like if a cysteine like amino acid isn't near another one, is it just gonna kind of like hang out on the outside?
Yeah, so because of that sulfur on the cysteine, they I guess it's categorized as a non-polar amino acid because of the covalent interaction it forms. It is a more polar non-polar amino acid, if you understand what I mean.
Yeah.
Because that sulfur has a lot of electrons. It is a very electronegative atom. So it is fine interacting in aqueous environments, though its primary function would be to form disulfide bridges.
Okay.
Yeah.
disulfide bridges, I remember hearing the video notes don't happen every single time.
They don't.
Do we know how to identify when they will happen and when they don't?
That's like too specific. You probably won't need to ever get into that much depth about disulfide bridges. It's just important to know that they are covalent. Yes. Yeah. And all the different types of bonds like dipole, ion-dipole, ionic bonds. They're all non-covalent interactions between our groups. I really wanted to touch the cysteine because that is a covalent one. I mean, this is very important. And it's like if you just happen to miss it, then it's like a very big thing that you're missing. All of the other R group interactions are non-covalent and they are all gonna vary. Ionic bond formations happen. Dipole, ion-dipole, all those that we talked about. They are not less important. They're just harder to miss.
And they do have like different levels of strength. Like they're not all equally as strong. So like an ion dipole would be weaker than an ion.
Would be stronger than ion dipole. Or I mean, it'd be stronger than a dipole. Yeah. Yes. I don't think so. The only type of question about amino acid R group interaction forming is when we get to enzyme substrate. She's gonna talk about binding pockets and then amino acids present in the binding pockets. But right now you shouldn't worry about that. Anything else? Yeah.
And then just to clarify, ionic bonding is that stronger than hydrogen bonding or it's hydrogen?
Yes, it is strong.
Okay.
Yeah. So ionic bonding only happens between your negative charged amino acids, which are glutamic acid and aspartic acid, and histidines.
There you go.
Okay, you should probably look this up, but hold on, I'm gonna get you in a second.
Yeah, ion just happens between the positives and the negatives.
Yeah. So
That's all you have to know. You don't have to know like which ones are
Arginine-
positive, which ones are negative.
arginines, histidines, and lysines are gonna interact ionically with aspartic acid and glutamic acid. Yeah. And you can determine which ones are which because the positively charged ones are very long. Like their R group chains are like very long, and so they're like if you don't have a thing written down, but you look at the structure, you can kind of see it. Yeah, and they have a positively charged nitrogen, which is how they get their positive charge.
Anything else? Okay, so quaternary structure is not the same thing as tertiary structure at all. Quaternary structure involves multiple different polypeptide chains coming together to fold as one protein. These are called subunits. So let's say polypeptide 1 and polypeptide 2 come together to fold as one protein. That's going to have quaternary structure, and it's going to be subunit A, subunit 1, subunit 2. Not all proteins will have quaternary structure. A lot of them that we'll see do have quaternary structure, but not all of them have to have quaternary structure. And these interactions happen between R groups as well. The same types of bonds that we see in tertiary structure, we also see in quaternary structure. But the only thing that I would take away from tertiary and quaternary structure is the difference between like subunits and domains. You only see subunits when a protein has quaternary structure.
No.
Yeah.
No.
Yeah.
No, subunits are just a polypeptide chain. And then domains are independently folded parts of a protein, of a subunit. You see, subunit is just a protein.
Okay,
So I
never
didn't know
mind.
which one was spelling.
No, it's okay.
Yeah, no. Yeah, so subunit is just a name for a singular polypeptide chain. Quaternary structure proteins have multiple subunits. Very important problem. And domains are independently folded portions of subunits. So the question, the problem set that has the antibody picture that has like the whole bunch of like writing on it. Each one of those like labels is a domain, and each one of the colored chains is its own protein. And so they come together into a quaternary structured protein. They are each their own subunit. Good. Yes.
So you go over what a domain is.
Yep. So a domain is an independently folded part of a subunit. And a subunit is just a polypeptide chain. It's fine. You're good.
Yeah.
Don't worry about it. You're fine.
Okay. Sorry. Is everybody good? Okay. Okay. Some more.
Oh, okay. So, like Jen was saying before, they're not the same thing. Like I just mentioned, quaternary structure are multiple polypeptide chains. Tertiary structure is a single, like she said before. Not every protein is gonna have quaternary structure. Every protein has tertiary structure. Or at least like a theoretical tertiary structure. It is if it's been denatured or if it's been broken or something, then it loses its ideal tertiary structure and it also loses function. But the important thing here is that every protein has its own tertiary structure, every protein, and then some proteins have quaternary structure. And so like we just mentioned, they're stabilized by covalent and non-covalent R group interactions. Also disulfide bridges are very prominent in quaternary structure as well, because it is the most stable R group bond. And so you get the different types of non-covalent interactions. And then we just mentioned right here van der Waals interaction as well between the non-polar groups. Hemoglobin is a great example of this. I don't know if there's a hemoglobin question in this problem set, but there are different subunits in hemoglobin, and the more non-polar amino acids all stick in the middle because they do not like to be exposed to water. When water gets to non-polar amino acids, they tend to clump up, and it's called the hydrophobic effect. And you lose protein structure, it becomes a mess. It's like when you get water and oil, and you get those little clumps up the top when you're like cooking pasta or something. If you're not using oil in your pasta, then you just don't know how to cook. But when you do cook, like if you're boiling water and you put oil on it, you'll see that they clump up together. That's because they're hydrophobic. They are not soluble in water. If that's a good like visual representation for you, go for it. It's just like another way of thinking about how they're gonna, come together and try to expose themselves the least amount possible to water. Yes?
I mean, in the problem set, like when they're like referring to globular proteins,
Uh-huh.
are they specifying quaternary or tertiary structure?
Is that like a specific question?
And I remember it. I'll get back to you if I want to find out.
Okay.
It's in question 5.
Yeah, I'll get back to you on that.
Okay.
Okay. Do you wanna take the next one? Cause it's domain and segment.
Yeah, I can.
Okay.
Yeah,
Sure.
so for that question is 5B. That was actually one of my exam questions when I took the class. You want to think about barrel-shaped proteins as pores, or like a pore in the membrane, so that like a gated ion channel, for example, is a barrel-shaped protein, because you have the nonpolar groups interacting with the cell membrane, for example, right? Because they are nonpolar, and then you have the inside are groups that are polar enough so that, like, let's say it's a calcium gated channel or it's like a potassium gated channel for like action potential in neurons, then the ions can actually go through. Or like if it's a water channel, then water can go through. Yeah, like a globular protein, only the outside ones would have polar charged amino acids, or not necessarily charged, it's polar, and then the inside of it, like hemoglobin, for example, will have its non-polar groups so that the protein doesn't lose structure. Yeah. And then just real quick, domains versus subunits. We were just talking about this. So a single chain can form local 3D structure, which is domains. So each one of those like big folds that you see right there are its own little domain. And so two or more separate chains or subunits can orient 3D space to give quaternary structure. What that basically is saying is you're going to get two different proteins. They're going to come together. The R groups are going to interact, and then they're going to be a singular protein. And if you want the definition of domain again, domain is a independently folded portion of a subunit. Questions, comments? What's it?
Okay, more on R group interactions and the bonds that they make. Okay, so different R groups are gonna make different types of interactions based on their chemistry and their structure. You have your amino acid chart, which she gives you on the exam,
On the exam, don't worry about it. We don't need to
Yeah.
memorize it. If you want to go to med school, then you will have to do a some point. For now, you're good.
So basically you just kind of have to know what types of bonds each section will make. That's what she wants you to know. And when I took this class, I would kind of just find an empty whiteboard and then like write down polar charged, polar uncharged, non-polar, and then I would write kind of bonds they'd make. So I don't know if that's a strategy anybody wants, but polar charged R groups are going to make those ionic bonds that we were talking about earlier. So positive charges are going to interact with negative charges and form ionic bonds, which are stronger than ion dipole bonds. And polar uncharged R groups will make those hydrogen bonds. These are the like dipoles that we see. They're not fully positively charged. They're not fully negatively charged. Their partial positives and partial negatives are going to attract each other and make these hydrogen bonds. Again, with the difference in electronegativity. And these are polar, so they'll usually like be on the outside of the protein. They're not going to be like hidden away. And then when you think of non-polar R groups, these are going to make your hydrophobic interactions and your van der Waals interactions. They're going to want to be kind of concealed from the aqueous environment of the protein. They're not going to want to interact with polar molecules or polar R groups. So they'll kind of be more concealed, more on the inside of proteins. And that's kind of all she wants you to know. She doesn't want you to like memorize every amino acid, just what kind of bonds that they'll make based on their chemistry.
Yep. Okay, so we're talking about the tertiary structure. This is just to reiterate, these are very, very important. They only happen in cysteines. disulfide bridges are extremely important, and I know it sounds like I'm just trying to have you guys hammer at home. I really am. This is very important. It is the only covalent bond. So if you're asked about this, you know, they are the strongest structure holders for 3D protein folding. The strongest, right? And so just very important to reiterate. I don't want to sound annoying about it. It's just if you do get asked about it, you should be able to say that these disulfide bridges are being covalently bonded and they are holding structure together because they are stronger than every other non-covalent bond.
Okay, this is kind of a summary of the levels of protein structure we've been talking about and the types of bonds that you see in all of them. So I'll just kind of go over them again. So your primary structure is just your sequence of amino acids, the order in which the amino acids go from the N terminus to the C terminus. And these bonds are covalent peptide bonds, and they happen between amino acids. Secondary structure is where you see your hydrogen bonds. You're not talking about your R groups. You have your alpha helices and your beta pleated sheets. In your alpha helix, they're positioned outwards to interact with the environment. In the beta pleated sheets, they are arranged up, arranged down, and so on and so forth. Tertiary structure, here you see your R group interactions, and then you see a variety of different bonds. Your only covalent bond is your disulfide bridges between the sulfur and cysteines. And you also have like hydrophobic interactions, van der Waals interactions, hydrogen bonds, anionic bonds, and then the ion dipole bonds that we mentioned earlier. And then quaternary structure, which is not seen in all proteins, but some proteins do have quaternary structure. And they are multiple polypeptide subunits. And the types of bonds that we see are the same as we see in tertiary structure. Any questions?
This is just a helpful chart. Your note sheet for the exam should have some sort of variation of this. It doesn't have to be this word by word. But some visual that helps you memorize the types of bonds present in each. You don't want to be talking about R group interactions if the question is about secondary structure. That will make you lose points. And so just some sort of version of this. Again, I want to clarify the note sheet should not be your only source of knowledge during the exam. Trust me, you will not find every answer you have on your note sheet. You just will not. And you'll just start wasting a lot of time and get nervous and think that everything's going wrong. The note sheet on the exam should just be a tool to help you find very specific little things that you might have forgotten, like a binding domain or something like that. Not for you to memorize completely and look for sentences and just waste time. Don't use your note sheet like that. It'll be not so helpful of a resource, and it is just there to like supplement you. It is not to be the primary source of your information material on the exam. So some sort of chart like this would be very beneficial. So right here, we just have a very cool little chart that talks about the hydrophilicity of different R groups. You might be weirded out by some of the non-polar amino acids being farther down than some of the polar amino acids. This has different reasonings. So if you take a look at like a tyrosine, for example, right? It's the one that has like a phenyl group and it has an OH group. It is more hydrophobic, for example, than a serine. Now, you might ask me, John, why is a serine that's like a very small, like polar amino acid, like why is it more hydrophobic than that? And then I'll tell you that sterics really matter, right? So if you think about the size of a tyrosine, that's a big boy amino acid. It's not as big as tryptophan, right? But it is a pretty big amino acid. You need to think about these different R group interactions, and you need to think about how these are going to affect their interactions with other R groups and with other molecules present in the cell environment. After all, this is a cell and molecular class, and you should be also thinking about things happening in vivo. Yes?
I was getting this with the toleration of the interactions of amino acids actually gained about the
Yeah, okay. And yeah, this is just helpful. Laura likes mentioning glycine a lot. like you might think of glycine as like, oh, it's like the first non-polar one in like every chart, and it's like hydrogen. The reason why it is farther down and it is less hydrophobic than, let's say, an isoleucine is because that hydrogen not only has some sort of electronegativity, it is a pretty small molecule. It only has a hydrogen. Hydrogen is not that big of an atom, and it is easily able to interact with other R groups in order to make different types of non-covalent interactions. And so that is why it might be present in like a more aqueous environment than, let's say, the isoleucine. So yeah. Go ahead. All right. Got it.
Okay, this is kind of what John was talking about like a second ago. Sterics matter. The size of your R group is going to matter in terms of how it's going to interact with the environment. So size matters almost, if not as much as chemistry. You have to think about like where, so like phenylamine and the benzene ring, it's a pretty big benzene ring. And it's, sorry, it's a lot bigger than glycine. So just think about like the structure and how it's going to affect like its environment and its interaction with the environment. That's all I kind of have to say. What do you have to say?
Just talking about
The sterics, yeah. I just talked about sterics and size. Do you have
Okay.
anything to add?
yeah. So this out of this whole slide, I want to focus on proline. So proline is known as a helix breaker. So if you go to, actually no, it has drawn here. So proline's special in its own way because its R group actually ends up forming a cyclic ring with the nitrogen of the backbone. And so it kind of does not allow for the nitrogen hydrogen or for the hydrogen atoms bonded to the nitrogen to form interactions with other R groups, which then leads it to break helices as the name implies. And so if you're looking at one of those like nicely drawn like ribbon structure proteins that you might find on like whatever website you're looking at, the molecule that causes that curve on the alpha helix, because we're usually like one alpha helix up, and then there's like a low strand, and then like another alpha helix. There's proline there. Think about the structure of proline and why that happens. Think about the lack of hydrogen atoms present and why you can't be making these hydrogen bonds.
And this is just the amino acid chart.
You get this on the exam, don't worry about it. Also, something else that someone brought up to me during office hours. The positively charged one is not a continuation of the non-polar, and then the negatively charged one is not a continuation of the polar. Just wanted to clear that up. Non-polar is its own thing. All of these are all polar. Like everything that's not non-polar is polar. These just have charges. So these are ionic. Okay. So we got some practice problems for you guys, just like 3 of them. I want to keep you guys here too long. So, which is more likely to be found in the interior of a protein, phenylalanine and tyrosine, and why? Anybody brave?
Phenylalanine would be more towards the interior because it's non-polar, so or it's hydrophobic. It's chemistry, so it's more on the outside where it's like exposed to the danger zone, I guess.
Good answer. Yeah, you're right.
Okay. If a protein denatures, what level of structure is R broken? Why? Anybody? Yeah.
Ordinary or the tertiary structures because those have relatively stable bonds. Primary this kind of bonds, so the nature of pretty much
I said secondary. So. Okay. That was a good answer, but the way I thought of this question was protein denaturing involves the breaking of hydrogen bonds. And when you think of hydrogen bonds, the first thing should come to mind should be that secondary structure. Cause your backbone atoms are involved in hydrogen bonding. Your secondary structure, it should just be like ring a bell. Hydrogen bonds and denaturing is the breakage of those hydrogen bonds. So your secondary structure is gonna be broken here. And that's why.
So every secondary would be the one that denatures, nothing else going cold.
Everything after.
Yeah.
Secondary, tertiary, and or quaternary if that has it. So anything non-primary.
Yeah.
Unless there's like a cysteine or disulfide bridge. Cause that requires a lot of energy.
Well, then you're also assuming that there's enough energy for that.
Right. Yes.
Okay, number 3. Mutation occurs that changes alanine to valine. What levels of structure are affected? Anybody? Yeah. Yes, but you're missing one. Nope. Oh, yeah, primary. Yeah, I mean, you could argue that they are small amino acids for the sake of, it complete changing. Yes, you could argue that it won't change the tertiary structure a whole lot. The emphasis of the question was to trick you into not saying it first for primary structure. But you could argue either way. You could say, oh, like the amino acid properties are similar. The odds of secondary structure breaking non-existent, and then you give a reason why. But then you can also say, oh, they changed, therefore it does change it, therefore I'm gonna stick with this, and that also works. So it's just it's a matter of like how you're wording it and why you're justifying it. But yeah, again, primary structure is gonna change. Primary structure is literally just a sequence. If you change the sequence, you're changing structure.
Yeah.
And then how about alanine to asparagine? Come on, guys, be brave. Yeah. Why?
Well, when amino acids can change, so that's primary. Asparaginine has, it's polar instead of being non-polar. Is that how it interacts with tertiary bond? And then so it's just-
Yeah, I could make that argument. It doesn't, but yeah, for instance, it's primary, tertiary, and or quaternary if it has it. If you really want to think about it like in a more technical way, then you have a polar amino acid in the middle of other non-polar amino acids, drawing this into water, and you could this lead to like stuff that we will learn further down the semester. But you just want to think about how non-polar amino acids are interacting with increased environments, and that would be very bad.
Basically, I would say like primary structure is definitely going to change, but the other ones like tertiary and quaternary might change. And
Probably.
that's how I would answer it. And then protein modeling. How to model protein. So I'll just do like 2-2.
You could do it.
Okay. So there are 4 different ways to model a protein. We have ball and stick model. Here, the balls are the atoms, and then the stick represents the bond. And it shows every atom and every bond with the backbone and the R groups. So it kind of shows you everything. And then wireframe, which is another model, just depicts those bonds without the balls. So it's just the wires, just the, no atoms. And then the spaceflow model is kind of like the ones that you see that are like bulkier and bigger. They literally take up like the space. It shows every atom in the space that it takes up and kind of shows like the shape. And you can't really see like each individual little bond like you can see in the wireframe and the ball and stick. This is more like just the overall 3D, yeah. Ribbon model, I feel like this is like super common. You see secondary structure, you don't see like each individual atom with their bonds. You just kind of see the overall, like you see alpha helices and you see beta pleated sheets and those kind of secondary structure of the molecule, of the protein, instead of each individual bond. And you can't see the R groups. They're most like for secondary structure. And then the backbone model only shows the backbone without the R groups and without R group interactions and like the skeleton, like the outline of the protein. And you can do like you can have different types of models in one protein. So you can model a protein with like 2 or 3 different ways just in one drawing.
I think the most common for visualization is ribbon.
Yeah.
I also think ribbon is the best to see like everything. Because you're less concerned about like R group interactions, and you're more concerned about the overall like structure that needs to function of the protein. And I think it's just more common.
Yeah, just because none of them like actually will like show like the R group interactions, but you can kind of like discern like if they're near each other or
Yeah.
whatever. Okay.
Yeah.
So that's just
Yeah,
the
look at it real quick.
depictions. Clearly ribbon is the best one. Like, don't think that it's weird, but yeah.
It's the fun one.
Alright, so that's it. Questions, we're gonna stick around to answer some questions that you guys might have. These will not be on the recording. So just get them answered right now. Alright, thank you. Stop recording.